pues empezamos, si os parece. Cambio el inglés y te doy paso. Well, good morning, everyone. Thank you so much for being here another Friday. We have today a talk about molecular printing at the Micro and Nanotools group. And without further ado, José Antonio Plaza will introduce the speaker. So, José Antonio, thank you.
Thank you, Isabella. It's a pleasure for me to introduce Juan Pablo. Dr. Agusil received his biomedical engineer degree from the Universidad Iberoamericana in 2009. He obtained an IBEC scholarship to study his master's degree at the Department of Physics from the Universidad de Barcelona in 2010. And in 2015, he finished his PhD in nanoscience at the same university. And the PhD was focused on the chemical functionalization of surfaces and suspended chips for applications in microfluidics and biosensors. Later, in 2015, he joined our group, the Micro Nanotools group at CNM, and he has been working on the development of suspended microtools for barcoding and for living cell applications, with a special focus on the surface modification and liberation of silicon-based and polymeric suspended chips. Juan Pablo is a fundamental piece in our group. As a biomedical engineer, he's the connection between the biological and the physical part of our multidisciplinary projects. Lastly, I want to say that he's also an excellent person, which made it easy to work with him. So now I think Juan Pablo is going to present more or less his activities during these years. And more important, he is going to present you all the techniques that we can offer to you in case you are interested in collaborate in these topics with us. So Juan Pablo, when you want, please.
Thank you very much for this very nice introduction. So I'm just going to share my screen. Can you see my screen? Oh.
Yes.
Okay, thank you very much. Good afternoon, everyone. Well, as without further ado, I will try to explain my part in the Micro and Nanotools group. And first, we are starting with the fabrication or the understanding of active surfaces and microparticles. Usually, these surfaces are modified with certain kind of biomolecular or quantum-based or nanoparticles to create different applications in their biointerface for sensing, transduction, or actuation. Why do we need these kind of surfaces? Well, because each of these surfaces have a certain application in the future as sample extraction or handling to create biointerface with cells, or as sensing to detect physical chemical changes, or transduction is to transmit certain information, or in actuation as delivery or anti-counterfeiting or barcoding. The idea is to adapt these inks on top of our smart surfaces. And throughout the time, there are two ways to fabricate these kind of surfaces. So you can build the surface with the functionality, or you can create a posterior functionalization. In a built-in functionality, like in the example of these microparticles, the same material in which the microparticle is fabricated is the one that offers the characteristic that you need. On the other hand, you can take a blank microparticle and then try to include all this functionalization in different parts of the microparticle or your surface. In this case, you have to take into account two important things. First is the anisotropy of this microparticle, or the use of, the use of orthogonal chemistries. But what is anisotropy? Well, it's a property of a substance to exhibit variations in properties along different functionalities. What it means is that it depends how you measure, it depends on the outcome, depends on how you measure the certain microparticle or substrate. So in our group, we've identified four main anisotropies: physical, chemical, surface coverage, and surface partner anisotropies. Each of these properties or anisotropies offer different properties to your substrate or microparticle. In this case, This is limited to microparticles, but you can take several of these cues into your surfaces, like for example, their shape, their size, or the topography. More interesting is a chemical anisotropy, which includes all these functional probes or nanoparticles that are covalently bound to the surface. Then, thanks to the advancement in technology, we can create these chemical anisotropies in certain areas of your substrate, creating certain patterns in 0, 1, 2, or 3D formats. And finally, in this case, in microparticles or also in small substrate, you can pattern the top or the bottom surfaces. So these four main anisotropies build what we call the universe that creates the most customizable surface or particles for your own uses. But in this case, for the use of this presentation, we're only going to use these three: chemical, surface, and surface coverage anisotropy, to develop a functionalization strategy for your substrate or your microparticle. So that's why the topic of this talk is molecular printing at the micro and nano scale, from flat substrate and microparticle functionalization. We have developed content printing techniques because they offer a lot of advantages for microarray or surface or microparticle functionalization. For example, they offer polymer printing. You can use biomolecular inks or nanoparticles, and you can multiplex. That means different areas of your substrate with different kind of inks. And the best advantage or the best technology to create these functionalities is using sublithography. Sublithography was developed almost 30 years ago with the introduction of microcontact printing. It's a pretty straightforward technology. You have a master with the shape of the structures that you want to obtain, and you replicate this shape with a mold. Usually we use an elastomeric stamp. This stamp then is inked with the biomolecule or nanoparticle that you choose, which is usually in a solution. And then by contacting a desired area of a substrate, you can translate the design of your master with the ink that you use as in inking. This technology offers a lot of advantages, as exemplified in this work, is that you have a lot of designs in a single stamp. You can design the mold here in the CNM with photolithography. Then you can create all these, in a single stamp, you have a lot of designs, and the designs can go down to the micrometer size features. But then all the advantage of this technique is that you can create very large areas to functionalize. In this case, you have 6 centimeters by 2 centimeters, which in the micro scale is huge. And moreover, it is important to have all this area or this functionalization, which is homogeneous. So this technology offers homogeneous patterning along the surface. But in the case that you require to go a little bit deeper and create smaller features, there's an alternative, which is called Polymer Pen Lithography, and it was developed by Chad Mirkin in 2008 and his group. And it followed the same principle as microcontact printing, but in this case, it uses a polymeric stamp that ends in sharp features. In this case, these pyramids. You ink these inverted pyramids, and then you can contact the area of the substrate that you are interested, and you only transfer a small portion of your ink. This kind of technology requires a special equipment, but it helps you create complex designs by repeated printings, just by moving the stamp. And more importantly, you can create sub-micrometric sized features. Since it is a pyramid stamp, and if you press hard on one side of the stamp, you can create different sizes with the same stamp. So you have more ink or more of your substrate, which is functionalized with more of the ink that you are interested. And since it is a repeated printing process, you can create a lot of different densities, a lot in single substrate. So we have been using these techniques to translate functionality into several types of substrates and microparticles in the group. We use microcontact printing in the Micro and Nanotools group. And the first part that we developed is an immobilization strategy. We have used silicon dioxide, SU-8, and glass. And the idea is to generate a reactive surface. In this case, we can call it a chemical glue. This chemical glue will react with the ink that you want to attach to your surface. In this case, we can use an epoxide silane to create active surfaces to attach thiols and amine groups, or an amine silane to create an hydroxysuccinated ester reaction site. And that way you can create active areas on top of your original pristine substrate. Following this strategy, and in our case, SU-8, we developed microparticles that were initially attached to a substrate, and then we functionalized, in this case, with two fluorophores, daylight 550 and daylight 488. The microparticles were printed with microcontact printing and then subsequently released. The image that you see is a collection of a lot of these microparticles that were functionalized and then released with fluorescent microscopy. Then again, we can follow the same procedure to attach proteins or other kind of biomolecules like DNA, which both of these proteins exhibit fluorescence, either naturally, like the green fluorescent protein in the center image, or with a fluorophore dye, which is attached to the DNA probe. Moreover, we can use the same technique to add quantum dots of different wavelengths to these microparticles. If you see, the shapes of the microparticles are the ones that were shown previously, but in this case, the images remain still on the substrate. These ones can also be liberated, but as you can see, it's a homogeneous procedure to create functional, or in this case, tagged microparticles along the substrate. If you can imagine, these microparticles are raised. So if you have a flat substrate, you would just obtain a homogeneous modification of your substrate. We can liberate these microparticles, and it's a robust covalent bond which maintains the quantum dots on its original position. We can characterize this, in this case, is the microparticles, but we can do it with a flat substrate. You can see in the image, or in the microparticle on the left, which is all coated with quantum dots, while the underside of the image on the right has not a single quantum dot. So we can characterize and see the density of the printing of the quantum dots. Moreover, these kind of microparticles were used as tagging in live cell experiments, in this case some HeLa cells, and you can see here in the arrows it points where these SU-8 microparticles with quantum dots ended up inside living cells. The cells are just marked green in the tubulin, the microtubules, and red in violet in the actin filaments. But it's just to get an idea that even in the harsh-ish environment inside a cell, these microparticles remain coated. As a lucky part of this experiment is that we can create lucky, because it is not intended to be like this, but we can obtain this kind of functionality out of Intaglio transfer printing. In this case, is the design that you want is not in your stamp, it's on your substrate. In this case, shown in this paper is the Intaglio trench. When you have a stamp that is loaded with quantum dots, and then you press it against this trench, only the design that is left on top of the trench will remain, and then you can transfer that design onto your substrate. So this is, let's call, the other side of the coin of the microcontact printing of quantum dots. And we can create active areas which are not coated with quantum dots, in this case, with all these designs that we had for the microparticles. And recently, this kind of technology has been very interesting for the creation of, for example, pixels for the new generation of flat TV screens. And they use this, they remove part of the quantum dots that they don't want, and they print the one that remains on the stamp. So this is another very interesting application of this microcontact printing. But in the case, if you want to go a little bit deeper to create smaller features on the substrate, well, we have a polymer pen lithography machine in the group. This prototype was developed with along several national projects, and it is basically a fluorescent microscope which has a sample with that holds a stage with your sample that has a 6 degrees of freedom. That means that it moves in X and Y and Z, and then in yaw, pitch, and roll. With these kind of movements, we have micro positioners for the micro scale, and then a nano positioner for a perfect alignment at the nano scale. This system helps you localize the stamp on top of your substrate on the desired area. The stamp, as I showed you previously, it's inverted pyramids, and we've characterized these inverted pyramids so we can tune the size of the spots that we want along all the surface. In this case, we have sharp pyramids that are coated with ink and then are transferred to the substrate. Moreover, we studied the behavior of these pyramids under pressure. So as you can see in the image on the left, the different deformation of the pyramid generates different contact areas, which translates into different printed features. The simulation that was done between the 2 green lines on the right graph shows the average Young modulus of the PDMS, which is the elastomer material that we are using to fabricate our stamps. And you can see the red and black points in the curves that show the correct size along the force when we print with our machine. So we can generate features from 1 micron down to around 790 nanometers per printed spot. Taking into account the micro and nano positioners of our machine, we can create multiplexed polymer pen lithography designs. In this case, for example, this protein with germ agglutinin, which was first modified with Texas red to emit in red. Then we translated the stamp, which was loaded with a green protein, with a green emission protein. And then finally, we translated the stamp along the substrate, and we added a third biomolecule, in this case, an antibody. And if we join the images, you can see that we have a multiplexed microarray generated on top of the substrate. It is interesting to see that every single let's see, unit of printing, it's done in a 3 by 3 square. So in the insert on the top right corner, you have a square that is less than 20 microns. Every single collection of spots is within a 3 by 3 square. So we were interested in to see if this kind of arrays remain functional or at least detectable by biomedical processes or biological processes. So we use the same approach to fabricate with germ agglutinin protein array with spot diameters of around 890 nanometers with the difference with this kind of distance between the spots. And then we just try to investigate if it was going to be recognized by antibodies. And we could see that the printed proteins remain recognizable by antibodies. If you see the overlay image in here, the antibodies selectively graph onto the printed proteins, what helps us guarantee that these proteins remain at least recognizable by the antibodies for future protein array applications. What I didn't tell you, but it was what is hard in this kind of application, is that these printed proteins weren't done in a flat substrate. They were done in this array of raised platforms. So the important part of the machine is to align the pyramid stamps with each of these raised platforms. So that's why it is important to have the submicrometer positioning with a precise substrate and stamp alignment, and the precise force control so you don't break this kind of substrate. That is the importance of having a very precise machine. And well, out of this work, we were able to collaborate and develop a startup, which is called Array for Cell Nano Devices, which basically uses this kind of arrays when the platforms are broken and then inserted inside living cells to track, well, different intracellular mechanisms. So this technology has been used for this company. Moreover, following the quantum dot applications that are quite relevant at this moment, we can also create one of the first quantum dot polymer pen lithography, this case on a flat substrate. And as you can see, the dense printing spot has a diameter of approximately 750 nanometers, generating discrete areas where the quantum dots are located. And following that strategy, you can create structures like this one with three different emissions, quantum dot with emissions, in this case, green, yellow, and red, generated a functional or at least identifiable substrate with a unique color code. So we use this kind of technology to create suspended SU-8 microparticles, which can be functionalized with these tiny little dots, spots of quantum dots to generate coded microparticles. In this case, as you can see, each of these microparticles has 1, 2, 3, 4, up to 6 different spots to generate a uniquely coded microparticle. And going a little bit further into the functionalization strategies and the things that we have and the possibilities that we have to functionalize in our group, is that we use the combination of polymer pen lithography to generate fluorophore patterns as a code and microcontact printing of quantum dot to generate a tag. In this case, we have SU-8 microparticles, where the top side of the microparticle is printed with the fluorophores, and then the underside are microcontact printed with a quantum dot. So if you select certain microparticles and you define a tertiary system, you can create a lot of coded microparticles in parallel with a single printing application. And we're lucky to publish all this information. And the important part of this application is that we are some of the first groups to combine both microcontact printing and polymer pen lithography to obtain precise alignment and modification of different substrates, whether it's a flat surface or a microparticle. So in summary, we have developed a customizable immobilization strategies to attach covalently attach different or multiple biomolecular and quantum dose-based inks in silicon, silicon dioxide, SU-8, and glass, obtaining micrometric and submicrometric patterns along these substrates with the precise substrate and alignment required for this procedure, and without breaking any of these of the substrates. So this quick presentation was, well, we are ready to innovate and collaborate to whoever thinks we can offer you a solution for this, for your problem with this kind of application. And we are very open to talk to anyone who has any doubt or any interest with this application, and hopefully collaborating in bigger projects. So thank you very much for your attention.
Thank you, Juan Pablo. It was a really good presentation. Thank you very much for your time. And now it's the time for the Q&A. We have Juan Pablo here. We also have Jose Antonio. So if you have any question or collaboration proposal, go ahead and ask the leading people. You know, you can write them on the chat and we will read them out loud, or you can open up the microphones. If there is anybody, I will start by asking if there is any current collaboration going on within the CNM.
Right now, within the CNM, we don't have any single collaboration. We have worked with groups in Madrid and in the UK, in the University of Bath. But right now, here in the CNM, we haven't done it.
Okay. So hopefully we start one today.
That would be great.
So, hi, it's Cesar. I've got a question. So first of all, thanks for your presentation, Juan Pablo. It has been very nice presentation, very well illustrated with all these pictures, very nice pictures of the fluorescence of the different quantum dots and molecules that you've immobilized on your particles. I've got a question about the release of the microparticles once they've been functionalized. Did you have any limitations just to keep the functionality of the quantum dots, but also of the biomolecules that you immobilize on your microparticles?
Well, thank you for the question. It's a very interesting question, and we were fighting quite a lot with that at the beginning of this project. Luckily, we found a way to release these microparticles using it's a mechanical force that uses a membrane that is first solidified on top of the microparticles. This membrane is it's a commercial membrane. It's called fluoromount. It is actually used to prepare microscope samples. But in a serendipitous accident, we found out that if you leave it to solidify at room temperature, you can create a membrane that protects surrounds the microparticle along the functionality, the functionalization, and then you can just peel it out of your wafer or your substrate without damaging either the microparticle or the top printing.
Okay, that's very nice. Thank you. And just another question about because you've been functionalizing your microparticles just for encoding them, but also I guess that your aim is also to use them like for interacting with some, I don't know, biomolecules or structures that you may have inside the cells. So have you worked on that, or perhaps I missed something during your presentation and you've already said something about this?
No, the thing is that we have this kind of work. We have a paper in collaboration with the CIB in Madrid, in which we actually took these microparticles, the ones that were eventually the ones developed for the company, and they were introduced into living cells to understand how pH changes inside living cells.
Okay.
So we have a paper. It's Advanced Materials 2016, and then you can all the information it's presented there. Or if you want to talk it, I can explain to you in person.
Okay. So thanks very much.
Thank you.
Thank you, Cesar. I think there is another question now. From Luis, yes. I was.
Yeah. Hello. This is Luis. As always, I have a few questions just out of curiosity, because I'm not at all familiar with the techniques you have been explained to us, soft lithography of whatever. So how hard is to get this stamp? So I assume you make the master and then you make this polymer stamp. Is it easy, and is it a disposable element? So once you have used it for one ink, you need to dispose of it and have another one ready?
Okay. Your first question, if they are difficult to fabricate? No, they're not. It's PDMS. So you just mix the polymer with the pre-polymer, and you cast this mixture on top of your mold. You can create molds here. For example, we have some silicone molds using it's photolithography, and then you use to etch the parts that you want to create, like the protruding features of your stamp. And the only detail that you have to take into account is you have to create like a Teflon layer on top of this stamp, so the PDMS doesn't bind that permanently on top of your silicone. So you use a fluorosilane. We have it there, and if you go into the nano part of the clean room, you can find it. Or you can use SU-8. and do a spin coating of a thin layer SU-8 and then photolithography. And then you can just cast this PDMS and replicate the mold. Then on your second question, are they reusable? It depends. In my experience, if you're going to use the same protein over and over and over again, you can have an extended life of this stamp. What you have to understand is also it's a polymer, so it can get damaged easily. And if you sometimes need to PDMS, it's a very hydrophobic. And if you want to print something that is in a water-based solution or something that is hydrophilic, it is usually activated in plasma, in oxygen plasma, in the TEPLA basically. But this creates, the longer you use it, it creates certain cracks on the surface of the PDMS, which can damage your final pattern. But if you're just using ethanol-based solutions or something like that, you can use it quite a lot. But then again, it's just PDMS. You buy the 1 kilo bottle and you can have all the replicas that you need.
For example, when you showed us this multiplexing of the 3 dots with different colors, so each color was done using the same stamp, or you change it?
Well, in this case, it would be a little bit more difficult to start changing the stamp. So what we did was a very long stamp, and we divided it into areas. And then so you have to align it only once instead of taking it out and then align it again.
Yeah, much clever.
The problem is that with this technique is you have to be very precise at the beginning, because you cannot fail. I mean, you cannot like touch and then, oh no, I made a mistake, do it again, because you already transferred the ink. So it's like a one and only chance.
The pyramids in the master, do you prepare them by KOH?
Exactly.
Okay.
Yeah, Marta helps us with that.
Oh, perfect. And at the end, you showed some particles that were contact printed in both sides. So I guess that in the first go, they may be at wafer level with preformed particles, but then the second part, you did it on the microparticle itself, no?
Yeah, it was also a serendipitous accident, but it was a very interesting one. These are SU-8 microparticles, which were produced by photolithography in a silicon wafer. So we don't have the food that was found in the silicon in the raised platform. These were directly on top of the silicon. So first we printed on top of the microparticles, and then we used the same release method with this membrane. But the good thing about this release method is that it leaves the microparticle that was on top of this is the substrate, the microparticle was here, and then you have nothing. The membrane goes around the microparticle, and when you peel the membrane, you have a flush, it's a complete flat surface of the underside of the microparticle still in this membrane.
Yeah, so the membrane becomes the second substrate for the second iteration. Okay.
And then you can just print on top of it.
Okay, thank you very much, Juan Pablo.
No, thank you.
Thank you, Luis. There's a question on the chat. Ferran says, maybe I missed it, but the PDMS stamp is chemically functionalized before attaching the ink?
No, I prefer to use like pristine PDMS for several reasons, because usually when you have to, as I said, if you want to use aqueous inks, you have to activate the surface so it can generate these other complications. But also to functionalize it, you have to activate it first. So it happens, you have the same risk of creating these fractures on top of the PDMS if you are just activating the surface to create these hydroxide groups. That's our OH groups on the surface of the PDMS. So it is reactive to a certain other molecule that changes the structure of the surface. Changing the structure of the surface can make it more hydrophilic, or basically hydrophilic, or that it attracts certain molecules stronger or weaker, so you can transfer to the substrate. So functionalization, it works. I've used functionalized PDMS, but I prefer to use pristine one.
Thank you. I don't think there is anybody in line now for another question. So last chance.
I have just a philosophical comment on your categorization of anisotropies. There was the one in the bottom, that it was top or bottom, no? According to what you have shown to us, shouldn't be single side or double side, because for me, top and bottom are symmetrical. So I don't see much difference regarding the behavior that you may expect of the microparticle. I don't know if I have explained myself.
No, it's an interesting comment. Yes, the thing is that the top and bottom was, we were trying to make it like as simple as for the reader to understand that when you are functionalizing, when it's in a wafer is top, and when you functionalize in the membrane is bottom. But we all, in the end, we just call them Janus microparticles. And you know, Janus is this, I think, Roman, Greek god that has two faces. So you have Janus microparticles. They call it, well, one side or the other side, or top or bottom, or, well, top side, underside. Yeah.
Okay, thank you.
No, thank you.
Thank you. But I also said thanks. Very nice talk. Okay, so if there isn't any other question, we will close for the day. Thank you very much for attending the presentation. Thank you, Juan Pablo, for doing it, and Antonio for introducing it. That's it for today. Next week, we will have an invited talk with a researcher from the Artificial Intelligence Research Institute of the CSIC about artificial intelligence. So we will see you there, and have a good Friday and a good week.
Thank you very much.
Muchas
Thank
gracias.
you.
Thank you. Bye-bye.
Bye-bye.
Bye.